This increased fatty acid synthesis function as a source of cytosolic acetyl coenzyme A and additional source of NADPH. How it function as a source of cytosolic acetyl coenzyme A? That pyruvate enters mitochondria, producing acetyl coenzyme A, and which combines with oxaloacetate producing citrate. And this citrate leaves mitochondria, enters cytosol, and cleaved by ATP citrate lyase induced by insulin, and it is broken down to acetyl coenzyme A and oxaloacetate. In that way, we get cytosolic acetyl coenzyme A. Now, increased TAG synthesis, that is triacylglycerol. Fatty acyl coenzyme A is available from de novo synthesis and from chylomicron, and which is absorbed from the intestine, and this glycerol 3 phosphate is provided by glycolysis for TAG synthesis, and this TAG formed in liver, packaged in VLDL, secreted in blood, used in adipose and muscle tissues. What about amino acid metabolism in liver? That is here increased amino acid degradation because amino acid is not stored. Increased amino acid during absorptive state, liver cannot use that much amino acid, and amino acid cannot be stored. It is either used for protein synthesis or degraded and deaminated and carbon skeleton degraded to pyruvate acetyl coenzyme A or TCA cycle intermediates. And the other way, branched chain amino acids, leucine, isoleucine, and valine are metabolized in muscles and utilized for energy. Increased protein synthesis during absorptive state replaces degraded protein during previous fasting period. Now we discuss about adipose tissue, that is energy storage depot. It is second only to liver in distributing fuel molecules. 70 kg man, white adipose tissue, 14 kg or half the total muscle mass. What happens to carbohydrate metabolism in adipose tissue? There is increased glucose transport by GLUT4, and glucose is phosphorylated by hexokinase. Increased glycolysis, and that leads to increased glycerol 3 phosphate, and that leads to increased synthesis of TAG. Adipose tissue lacks glycerol kinase, that is why it is not degraded here. Increased pentose phosphate pathway activity, glucose metabolized through producing through this PPP path, this pentose phosphate pathway, and produces NADPH, which is essential for fatty acid synthesis. Now about fat metabolism, fatty acids from exogenous TAG added to adipocytes TAG and stored, and endogenous TAG comes out of liver as VLDL. This fatty acid is released from lipoproteins by lipoprotein lipase. In adipose tissues, lipoprotein lipase is upregulated by insulin, favoring storage of TAG during fed state. Insulin is high, then it upregulates this lipoprotein lipase, which releases the fatty acids from TAG, and that enters the adipose tissue where it again converted to TAG and stored. This elevated insulin favors dephosphorylated, that is inactive form of hormone sensitive lipase, and thus inhibiting the lipolysis in the well fed state. The hormone sensitive lipase breaks down this TAG, but when the insulin is high, it is inactive, and then lipolysis is by hormone sensitive lipase is inhibited. In resting skeletal muscle, that is the rest muscle is 40% of body mass, can use glucose, amino acids, fatty acids and ketone bodies as fuel, take up glucose via GLUT4 for energy and glycogen synthesis, and take up amino acids for energy and protein synthesis. No covalent regulation of PFK2 in skeletal muscle. That is why you know this no intracellular shows that increased glucose, but that way it is glycolysis is activated. Carbohydrate metabolism, that is increased glucose transport, carbohydrate rich meal, increased plasma glucose plus increased insulin, and glucose enters myocytes via GLUT4 and phosphorylated to G6P via GLUT4 by hexokinase and metabolized to produce energy. Increased glycogenesis, increased insulin and glucagon ratio and available G6P leads to glycogenesis, particularly if glycogen store is depleted as a result of exercise. And what happens to fat metabolism in skeletal muscles? Fatty acids released from chylomicrons and VLDL by lipoprotein lipase, but glucose is the primary source of energy for resting muscle during fed state. Fatty acid is a secondary option. Amino acid metabolism, increased protein synthesis, that is increased amino acid, and that leads to increased protein synthesis. The next one is increased branch chain amino acid uptake by skeletal muscle, that is a principal site for branch chain amino acid degradation, and which are having required transaminases. Now the dietary branch chain amino acids pass through liver and taken up by muscle for protein synthesis and energy. Brain, that is 2% of adult weight, 20% of basal oxygen consumption of the body at rest. You know how much oxygen it requires. In fed state, brain exclusively uses glucose as fuel through GLUT1 or blood-brain barrier vessels, and which is insulin dependent process, and that completely oxidizes glucose to CO2 plus H2O. No glycogen, no TAG stored in brain, so depends only on the blood glucose. If blood glucose level is less than 50 milligram per deciliter, then cerebral functions is impaired and compromised. Now you see what happens. Week 2 of quarantine, we are having discussion, we are having fighting, don't know what we are having, but we are all together in this corona pandemic, and we have to be united but socially distant and protecting ourselves and our family members. And you are in a home stay, but please don't eat like these people. Then you are going to having a great problem afterwards. After when colleges is going to open, then you will have difficulties in moving around. So eat healthy and be healthy and stay safe. Thank you. Here you see the mother is with food and one is having food and another is not, and he is in a fasting state. Next class we will discuss about the fasting state. Thank you again.